A groundbreaking discovery in molecular biology sheds light on PAANIB-1, a potential therapeutic treatment for Parkinson's disease, which is a neurological disorder marked by the gradual loss of dopamine neurons in the brain. It involves the C-gas sting pathway, a vital mechanism that detects damaged or suspicious DNA in the cell's cytoplasm and orchestrates a rapid microglial immune response as a result. This process is linked to parthenidose, a cell death mechanism to isolate damaged or infected cells and prevent the spread of disease. But when excessively engaged, these pathways can lead to severe inflammation and unintended widespread damage if the C-gas sting pathway reacts to large amounts of DNA fragments created through parthenidose. But why and how exactly does parthenidose occur? In Parkinson's, it begins with a protein called alpha-synuclein, which misfolds and accumulates over time to form damaging fibrils that create Lewy bodies. This disrupts normal cell function and causes severe oxidative stress on neurons. In response, a protein in the cell nucleus called poly-ADP ribose polymerase, or PARP1, typically orchestrates the repair of such stress-induced damage by creating poly-ADP ribose, or PAR, a scaffold to aid in DNA repair. But excess stress can cause an overproduction of PAR. And as the surplus ventures beyond the nucleus, PAR finds its way to the mitochondria, where it interacts with a key mitochondrial membrane protein known as apoptosis-inducing factor, or AIF. This interaction releases AIF into the cell's cytoplasm, setting it on a course back towards the nucleus. During its journey, AIF binds to a critical protein called macrophage migration inhibitory factor, or MIF. Once this complex reaches the nucleus, MIF's nuclease activity initiates DNA cleavage and fragmentation. The resulting DNA fragments escape into the cytoplasm and trigger new C-gas sting pathway activity, thus perpetuating a cycle of inflammation and stress. So how should intervention be approached? Traditionally, it has been believed that blocking the initial stages of parthenidose would be the most effective in preventing DNA damage and cell death. For example, current cancer therapies that target early steps of parthenidose have drawbacks, as they hinder essential DNA repair in the process. Therefore, let us shift focus downstream to try and directly prevent MIF nuclease from triggering parthenidose without interfering with important upstream functions. Exciting new research has unveiled PAANIB-1, a groundbreaking MIF inhibitor that stops the last step of parthenidose. PAANIB-1 blocks MIF's nuclease activity without interfering with its other functions, and prevents the generation of harmful DNA fragments. MIF's unique structure differentiates it from other nucleases in the body, thus ensuring selective inhibition and minimizing adverse effects. Targeting MIF using PAANIB-1 shows promising clinical potential, and there is hope for a future where the progression of neuronal damage and symptoms of Parkinson's disease could be halted or even reversed.